Hello everyone, today we'll be discussing our project on mRNA-loaded dendritic cells for prostate cancer immunotherapy. My name is Pratheeka and I'll be focusing on the biological considerations and regulatory standards. So let's get started with some background. Prostate cancer is one of the leading causes of cancer-related deaths in men. The current treatments often lack specificity in targeting cancer cells. This is where mRNA therapeutics come into play. They offer a targeted and innovative approach by instructing dendritic cells, or DCs, to activate the immune system against tumor-specific antigens. Our project focuses on exploring strategies to implement scalable, GMP-compliant processes for manufacturing RNA-loaded DCs, taking these therapies from a clinical phase to a commercial one. And then in phase 2 of this project, we expanded our work to include production strategies and alignment with regulatory standards. This section highlights the biological and manufacturing considerations essential for designing a process that effectively uses mRNA-loaded DCs to treat prostate cancer. The mechanism begins when mRNA encoding tumor-associated antigens such as PSA, PAP, survivin, and HTERT are introduced into dendritic cells. Inside the DCs, the mRNA directs the production of antigen proteins, which are then processed into peptide fragments. These fragments are then presented on the surface of DCs via major histocompatibility complex molecules. This presentation then activates cytotoxic T cells, enabling them to target and destroy cancer cells expressing those same antigens. The key target antigens include PSA and PAP, which are prostate-specific markers, as well as survivin, which promotes tumor survival, and HTRT, which supports tumor growth by maintaining telomere length. A major challenge with mRNA is that it's fragile and susceptible to rapid degradation. To address this, mRNA strands are engineered with protective nucleotide modifications. This enhances their intracellular longevity and protein production. Equally important is maintaining dendritic cell viability to ensure effective antigen presentation. Dendritic cells are cultured with interleukins IL-4 and GM-CSF, which improve their antigen capturing and presenting capabilities. To maintain optimal conditions for both mRNA and dendritic cells, we use wave bioreactors. These bioreactors provide controlled conditions, including 37 degrees Celsius, 5% CO2, and gentle rocking conditions, which promote oxygenation and nutrient mixing while reducing shear stress. And this also ensures the stability of mRNA and the health of dendritic cells, preserving their activity as antigen-presenting cells. The entire production process is conducted in sterile closed systems to comply with good manufacturing practices, or GMP, as previously stated in our background slide. And this ensures that there's batch safety and also prevents contamination and guarantees consistent therapeutic efficacy. These GMP standards are critical for clinical viability of mRNA-based cancer therapies.
My name is Gracie, and here is the overall production of mRNA-loaded DCs. The next few slides will detail the manufacturing process from analogous DC precursors called monocytes to mature DCs. First, isolation of desired cells and mRNA must occur. This collection is performed by leukapheresis, which obtains a large quantity of monocytes. This method has high efficiency and yield. After collection, a density gradient medium is inserted into the blood, which then undergoes centrifugation and separates into the layers, as seen in this figure. Due to the maximum amount of blood collected from the patient, when scaled up, this process will always be on a small laboratory scale. The components desired are within the red blood cells, RBCs, which contain APCs, PBMCs, and monocytes, which are within plasma. When PBMCs are successfully separated, a washing buffer can be inserted to isolate for use further down. This process is similar to another FDA-approved therapy, Siblucil T, which has recombinant protein and contains PAPs infused with GM-CSF. PBMCs have CD4 plus T and CD8 plus T cells and are activated using engineering leukocyte immunostimulatory, or enlist cells derived from tumor-associated antigens, TAs. This therapy will have multiple TAs within the DCs for more visibility. The next phase within DC production is transduction and transvection, where cells are loaded with mRNA. First, mRNA must be collected from the patient and will undergo the following processes: pDNA linearization, chromatography to separate DNA, in vitro transcription, chromatography to separate mRNA, enzymatic capping, chromatography and filtration, encapsulation, and final sterile filtration to create the final mRNA. This method of creation is similar to Moderna and Pfizer. Next, the monocytes are collected from the patient's blood and grown into immature DCs with the presence of IL-4 and GM-CSF for around 5 to 6 days. This process is done in a rocker or wave bioreactor, which is effective for highly regulated cell cultures, such as the DCs. Scaling up issues do arise when needed to increase the reactor. However, viability of cells is shown to be 96%. After culturing, closed counterflow centrifugal illustration (CCE) for the highest purity and recovery can be performed to purify 83% of immature DCs. The last phase is to load cells with mRNA created to express specific antigens. Electroporation is quite common, which maintains cell viability by using GM-CSF to prevent premature cell death. When scaled up, this method has less than 2% loss of viability due to the incorporation of a thin, even slab with continuous flow and electrodes on both sides. This allows even spread of high voltage, which creates pores that allow mRNA to enter. Finally, IDCs are further activated into mature DCs within a wave bioreactor and similar culturing conditions as monocyte culturing. This is to help increase the expression of specific antigens and increase migratory capacity within the body. Maturation is effective with 98% viability and 71% yield. The next phase of the report follows the regulatory considerations and separation strategies within the manufacturing design. To recap the DC creation, first it starts with patient's blood collected with leukapheresis. It is then separated into the desired PPCM, APC, monocyte, and mRNA components through centrifugation. The mRNA is then taken for further creation for the loaded DC cells. While that is occurring, monocytes are cultured into IDCs in a wave bioreactor and then can be loaded with the mRNA created in the third step via electroporation. The loaded IDCs are then matured within a wave bioreactor. Things to consider are regulatory standards within the manufacturing aspects before moving on to separation and purification techniques. Finally, the production can be administered to humans for results.
So as previously stated, in phase 2 of this project, we focused on aligning these procedures with regulatory standards. GMP emphasizes sterile environments and closed systems, such as in the wave bioreactors, to ensure consistency in purity, stability, and mRNA integrity across batches. Additionally, rigorous analytical methods like high-performance liquid chromatography, or also known as HPLC, are used to separate, identify, and quantify components, verifying mRNA quality and encapsulation precision to ensure the product's safety and functionality. Preclinical evaluations assess the pharmacodynamic and pharmacokinetic profiles of the therapy. This includes identifying safe dosage levels, minimizing toxicity, and confirming the effectiveness of lipid nanoparticles, or LNPs, which encapsulate mRNA to protect it and enhance cellular uptake. Together, these evaluations ensure that the therapy is safe, effective, and ready for clinical trials.
To successfully administer this drug to patients, it must be effectively separated from its culture and purified. There are many methods that can be performed, but due to GMP all open methods must be eliminated due to the contamination risks. Therefore, centrifugation is the best method. The top layer pellets, which contains the pure DCs and can be washed with a buffer solution. However, some risks are that crucial cells could be washed out within the buffer solution, and decontamination must be performed after each centrifugation. This process takes around 4 hours due to the reassembly and decontamination of each chamber within the centrifuge. Next, checking if purification occurs through flow cytometry analysis. Unfortunately, this method is more expensive and the identification of CD4 plus T and CD8 plus T cells are redundant, but antibodies can be identified easily to ensure purity of DCs. The cells can be re-injected into the patient.
In conclusion, our project explored the exciting potential of mRNA-loaded dendritic cells as a next-generation immunotherapy for prostate cancer. This innovative approach harnesses the body's own immune system to target cancer cells more precisely and effectively, and it also addresses one of the leading causes of cancer-related deaths among men. Throughout this work, we've analyzed the biological considerations necessary to ensure the stability and viability as well as functionality of mRNA-loaded DCs. We also analyzed the manufacturing challenges in scaling up such therapies. And then in phase 2, we shifted our focus to the critical regulatory and compliance aspects, emphasizing the importance of good manufacturing practices and stringent quality control processes. So this study not only highlights the scientific and technical complexities of bringing mRNA-based therapies from research to real-world application, but it also underscores their potential to revolutionize cancer treatment. And by aligning innovative biological methods with robust manufacturing strategies, we can help pave the way for safer, scalable, and more effective treatments for patients in need. And that's it. This concludes our presentation. Thank you for your attention.
The following are the references from our 2 papers and the photos seen through this presentation. Thank you for listening.